Hello everyone. Let's continue with Heredity and Evolution chapter, class 10th. Last time we completed the homozygous, heterozygous organism. We completed about alleles. We studied about Mendel's experiment, how he carried out the first set of experiment, right? There are many things that we carried out more. So let's not waste the time for the previous content and let's start with the new content. Now, if I have to represent Mendel's experiment based on the homozygous, heterozygous, how can we name the allele as homozygous or how can we name the allele as heterozygous? So let's study it genotypically and phenotypically. Okay, how do we give the ratio of a genotype and a phenotype? Let's study accordingly. Fine. So Mendel, as we already know, that he took homozygous tall plant, which is represented as capital TTs, and homozygous dwarf plant, which is represented as small tt. Correct? I repeat, homozygous tall plant represented as capital TTs, homozygous dwarf plant represented as small tts. Correct? What did he do? Yes, he cross-pollinated these plants. Correct? And the result obtained was what? All the plants were tall as F1 generation. And that all the plants which were tall, rather than saying only tall plants, I would say it as what? Now how it is representing, the resultant is representing as how? Capital T and one small t, which says what? Which is a dominant trait, tall, right? So how do we say it as? Heterozygous tall plants. So all the F1 generation plants which was obtained in the first case when he cross-pollinated, it was heterozygous tall plants. Clear? Now, from this, as a question arises in Mendel's mind that I have taken one dwarf, I have taken one tall, I have taken one dwarf plant. Then where is the dwarfness characteristic in the plant, in the F1 generation? So what did he do? He took the F1 generation plant, that is heterozygous tall plant, and he self-pollinated the plant. Right? He took heterozygous tall plant and he self-pollinated the plant. What did he find when he self-pollinated the plant? What did he get the resultant plants as? Like out of 4 plants, he almost got 3 plants as tall and 1 as dwarf. So what is the conclusion here? And the F2 generation, what is the conclusion? That in F1 generation, the alleles which was not expressed, okay, the characteristics which was not expressed got expressed where? In the F2 generation. Correct? So when he self-pollinated, the resultant plants which were obtained was what? Capital TTs, 1, that is homozygous tall plant, 1 heterozygous tall plant, right, with a capital T and a small t, then homozygous dwarf plant with small t's, right, and heterozygous tall plant. Correct? So meaning what? These are the F2 generation plant that he got. Correct? So with this observation, Mendel concluded that even the dwarfness character which was present in F1 generation got expressed in F2 generation. Correct? So this was all about Mendel's first set of experiment. Now comes Mendel's second set of experiment. Now in the first set of experiment, the character that he taken was what? Was the height of the plant, tallness and the dwarfness. Now here in the second set of experiment, he took 2 characters, that is known as what? Dihybrid cross. Now when I talk about the first set of experiment, I would say it as a monohybrid cross. Only 1 character was taken. Whereas if I have to talk about dihybrid cross in the second set of experiment of Mendel, it is what? 2 characters are involved. One is the shape and second one is the color of the seed. The shape of the seed and the color of the seed. The shape of the seed would be round and wrinkled. Okay? The shape of the seed would be round and wrinkled. The color of the seed would be yellow and green. The color of the seed is yellow and green. So the 2 character dihybrid cross he has taken. The first one is what? About the shape, right? That is round and wrinkled. Second one is what? The color. That is green and yellow and green. Correct? Now, the shape of the seed is represented as what? Capital RR for round, okay, and small rr for wrinkled. And the color of the seed is represented as what? Capital YY is yellow and small yy is green. So he took homozygous round and yellow seeds. Now homozygous means what? Same alleles. Round, capital RR, yellow, capital YY. So he has taken what? Homozygous round and yellow seeds. And homozygous wrinkled and green seeds. Wrinkled is represented as small rr. Green is represented as small yy. Correct? Yes or no? And he cross-pollinated it. Right? Now you will be having a question saying why the round is represented as capital RR whereas wrinkled is represented as small rr. Why not W? The reason is when you show it in the form of a flowchart, when you have to represent it, it would be a bit confusing when you cross-pollinate and when you see the resultant. That is the reason what we represent, the dominant trait we represent more, right? So the dominant in seed, in the shape of the seed is what? Round, and wrinkled is recessive. So round alphabet has been taken, that is capital RR, and wrinkled alphabet is small rr. In the same way for yellow and green, yellow is dominant over green. Correct? Next. So now he cross-pollinated these plants and the resultant obtained was what? Capital R, small r, capital Y, small y. So I would say this as what? Heterozygous plant. Yes or no? What would I say it as? Heterozygous round and yellow plants. Okay. Now, in the second generation, if I had to find out, he self-pollinated these plants from the F1 generation. And here, how is the resultant obtained now if I have to take capital R, capital Y, small y? I have to give it in the form of 4. That is capital R, capital Y, capital R, small r, capital Y, and small r, small y. So these are the 4 conditions wherein we can write R, R, Y, Y. Yes. Now, these all have to be cross-pollinated, self-pollinated. Now we have to draw a table which signifies all the 4 categories, how it can be cross-pollinated with each other. Yes. So the resultant obtained is what? One result would be round and yellow. Then other will be round and green. Next wrinkled and green. Then wrinkled and yellow. Now when I say round and yellow, it is what? Capital R, capital Y. When I say round and green, it is what? Capital R, small y. When I say wrinkled and green, it is small r, small y. When I say wrinkled and yellow, it is small r, capital Y. Correct? Now, the ratio, if I have to go with round and yellow, out of this table, from this table, I got round and yellow how many? 9. As you can see, in the first side, it is capital R, capital Y, capital R, capital Y. Right? So, if you just count the number of round and yellow seeds in the table, you will find it as what? 9. Next is round and green. If you find round and green, now the one which is counted already, the 9, that you will not count here. So the one that is not counted. So round and green, where you can find green, round and green, it's 3. Okay. Wrinkled and green, rather than that, I would go with wrinkled and yellow, it's 3. And then wrinkled and green, it's 1. So the ratio goes as what? 9:3:3:1. Clear? This is how the ratio was obtained for the Mendel's second set of experiment. Right? Now whenever you go with, it's not confusing, it's very easy if you go it properly. If you see the table properly, you just identify the characteristic properly, you will be able to recognize it, right? You'll be able to find out the 9:3:3:1 ratio. 9, whenever you get 9:1 ratio, is what? Round and yellow seeds. Now that round and yellow seeds you have already counted, you have to mark it differently, whether with an underline, whether with a square or a circle, etc. And then you have to find out with the next one, which is that it is the round and green, which comes with the 3 ratio. And that 3 ratio, you will take that apart and then you will start counting the other set, right? In the same way, you find out another 3 and then 1 ratio, right? Only one wrinkled and green seed is available. Yes or no? So this is the 9:3:3:1 ratio. Now that we have covered the first set of experiment and the second set of experiment, now the time comes to explain the principles of inheritance. Now I will explain you what was the observation that Mendel gave with these two set of experiments. Okay, the first observation was F1 generation displayed only one of the parental trait. Based on this, he gave principle of dominance, one of the principle of inheritance. Correct? I'll be explaining about principle of dominance as well properly. Second, the hidden trait in the F1 generation, where did it reappear? Reappeared unchanged in the F2 generation. Based on this, he gave what? Principle of segregation. Third, 4 types of plants were obtained in F2 generation in a dihybrid cross. What is a dihybrid cross? It is the second set of experiment where he got 9:3:3:1 ratio. Remember? Right? That is round and yellow seeds, round and green, wrinkled and green, wrinkled and yellow. Correct? Round and yellow, wrinkled and yellow, round and green, wrinkled and green. Okay? So this was the third. So 4 types of plants were obtained in F2 generation in dihybrid cross. Based on this, he gave principle of independent assortment. Right? Now, if I have to state the principle of inheritance, initially in the first video lecture I did say the 3 principle of inheritance are what? Principle of dominance, second principle of segregation, and third principle of independent assortment. Let's go into it. The first one, principle of dominance. Now, in heterozygous organisms, I hope you understand the word heterozygous, right? In the previous explanation, it has been taught. So, in heterozygous organisms, only one out of the 2 alleles expresses itself. That is the dominant trait, while the other remains hidden. That is the recessive trait. So, this is about principle of dominance. Correct? Second is principle of segregation. Now here each allele retains its distinct identity even though they remain together in an individual. They segregate only during gamete formation. Correct? The third one, principle of independent assortment. Now here during the gamete formation, segregation of alleles of one pair is independent of the segregation of the pair, that is the other pair, right? Now here this independent assortment can be explained in the second set of experiment which Mendel did, that is about the 9:3:3:1. Independently all the 4 characteristics were expressed, right? So that is how is stated as what? Principle of independent assortment. Clear? I hope you understood the video lecture about Mendel's first set of experiment, Mendel's second set of experiment, what did Mendel observe with these 2 set of experiment, and how did principle of inheritance, that is dominance, segregation and independent assortment, came into picture. Please study well, study sincerely children. Thank you so much.